manipulators. And then once the science swarm is done and they get all their treasures off the vehicle, we get the janitor job usually of cleaning all the muck out of the bottom of the boxes and making sure all the jars are clean and the filter media is clean, and putting all that stuff back together, putting the core tubes back together. Depends on the science how many are aboard. They're a little short-handed this year, so we try and help out.
Yeah.
Obviously don't want cross-contamination from, if there was a sponge in the box last time.
Well, as a scientist who's going to be looking at some of those sponges, Abby and I really appreciate that, so thank you.
Can you come down a bit on the delta?
Oh yeah, sorry. Yeah, we've this year we're using our shop vac that is usually reserved for our mill in the shop, so it's working quite good.
Oh, nice.
We used to use that, that big hose and siphon the water out and use that kind of as a vacuum. But the shop vac's a little quicker.
I'll have to keep that in mind for future. How am I going to clean this out sampling?
Yeah, we're thinking of buying a dedicated science shop vac. Gets pretty manky after, then you have to clean the shop vac out, right? But it's easier to take that over to the sink and open it up and clean it out.
Yeah. Spray a little excess ethanol in there and wipe it down to kind of sterilize everything.
Yeah, we don't go that far. I don't know if we should. Some of the scientists are really quite picky about, they'll come out and inspect and make sure everything's clean. And it's like some with the Niskins, for example. It's this whole ritual of cleaning the Niskin out before it goes in the water again.
I actually have several people, because I do a lot of the eDNA work, who ask me, oh, do you make sure your Niskins get really clean and sterilized before you use them again? And I always say, why? They're open all the way to the bottom.
That's what I always said, but I've got a very long-winded explanation of why that is not valid. But
sterile on deck lasts just until it lands in the water, and then it's no longer sterile.
I guess if you're going around to different locations, it's just like with your boat, you're supposed to clean the hull off, so you don't cross-contaminate.
Yeah.
Or camping.
I kind of figure that, yeah, the freshwater rinse probably takes care of a lot of that, and everything gets kind of freshwater rinsed because you don't want seawater sitting in anything over time, because that's just bad for all the gear.
Yeah, that's always our first task after the science team is done, is we get the hose out and get everything, just in case it hasn't dried out enough, everything gets a bath. It's really a pleasant task when it's 75 degrees on deck, but not so much when it's like 45 degrees.
Yeah.
Yeah.
Washing your car in the rain kind of thing.
Well, you're now to the part of the expedition where you should have more of those 75-degree and pleasant.
Yeah.
As opposed to the Olympic Coast end of the expedition, where we were all like, well, Olympic Coast in October, it's going to be a little exciting.
We were so spoiled last year in Hawaii, and between Hawaii and American Samoa. Our biggest complaint was the air conditioning wasn't keeping up.
Yeah, I think that's the air conditioning struggling to keep up may have been what done it for the old eDNA pump that died en route to American Samoa. So I ended up shipping a mini pump out there. And it worked, but it was very slow.
Oh, I seem to remember that. Or
Did you have you had a filtration device on the vehicle, did we?
It was for the wet lab.
Oh, I see.
Yeah. We might have a filtration device on the vehicle for you to try out in some future expedition. Where I'm involved with a lab that's designing an in situ filter that will take the eDNA sample and preserve it in situ that we are going to be trying out over the next couple of years. But
would that be
right now
like
we're
a
still in the
single shot?
the idea would be that it actually will have in the current design up to 3 filter cartridges.
Like a rotate through?
Yeah, it'll rotate through. So the idea is really designing it for things like for Liz Clark's AUV, so that since it can't trigger a Niskin bottle, how could you get an eDNA sample with it? So it allow those kind of remote vehicles to be able to take water samples.
So it has to be super lightweight and power efficient for AUV.
Yep, lightweight, power efficient, and we've been dealing with like how do you what buffer can you use to preserve samples deep underwater? Because the buffer we normally use in the wet lab up there won't work because it has a chemical called SDS in it that precipitates at low temperature. And the current temperature on the bottom is 1.7 degrees C, so we've been looking at other options. But honestly, I like the take the Niskin bottle and bring it to the surface option. You also get a lot of water there. That can be used for a lot of things.
Yeah, I'm surprised Nautilus doesn't have a Niskin rosette. I guess they map and when we can't dive instead of run the Niskin rosette down.
We do have an underway CTD, conductivity temperature depth, but obviously doesn't sample water. It's proxy
We have
measurements.
a what? Sorry?
Conductivity temperature depth probe.
Oh,
CTD.
yeah.
We have the expendable ones, the XBTs, but we also have an underway that we can retrieve. So we dunk that for mostly for sound speed measurements while we're mapping. But
for the multi-beam data, right? Or for the
yeah, so we could accurately plot.
A couple more rocks coming into view here. Has this been the deepest dive of this expedition so far, or did they make it deeper on Davidson?
I think this was deep.
Yeah.
We should have been washing that cable off on the way up.
Yeah. So I'm gonna the ship's almost complete its move. I think I'll hold there just so we don't breeze past if there is some terrain here. We'll be able to spend some time. Plus we're about 200 meters laid back, 150.
how do you clean the cable? Just stand on the deck with a hose and point it at the...
I usually rig up some kind of gizmo that sits up on the A-frame on the overboarding sheave and drenches the whole overboarding sheave, so it gets washed as it comes over the sheave, basically. Otherwise, it's kind of messy on the deck.
I think the next dive is fairly shallow. They're going up to check out a potential Petrale shoal ground.
Sorry, we're going to check out a what?
Potential Petrale shoal spawning ground is, I think, the plan if the dive goes tonight.
Yeah, we're gonna take a look at the weather and see.
I think that's what Lisa's discussing right now during the check-in call.
Yeah. People need to come down a bit.
Nice little
Sorry, I wonder why you're bouncing.
Outcrop of bamboos and black corals here.
Yeah. Is that a chrysogorgia there?
Oh, that might be. Yeah, I think that is. I'll put a little note for Tom that he gets to see a chrysogorgia on transect.
We're just about 50 meters out from the end of the transect.
Yeah, well, hopefully we'll get some more rocks then.
What is this thing coming up?
That looks like another big primnoid. Yeah, that's another big primnoid sea found, probably Narella. And I think another little chrysogorgid urboid, that might be a black coral there on the left.
Hmm.
One of the programs that I work with that I get actually the most samples from is NOAA's groundfish trawl survey. And it's a great source of samples up to 1,200 meters because they go coast wide, and I get all the coral and sponge bycatch. But you never trawl this deep, and so all the species that are this deep are kind of a hole in our DNA voucher library. We've been working with museums to get museum specimens, but being able to work with a team like you all to do dives and actually see the animals in situ and collect more of them is a great opportunity to expand our resources for these really deep water taxa.
5 meter pitches that are coming through there.
Yeah.
How many meters do we have left in the transect?
About 25.
25, okay. One of our scientists ashore is recommending we look around a little after this transect, and I concur.
Yeah, I have the ship held
Hopefully.
up, and we're still going to be inching through this, but not really quite sure which direction to go after that because we're going to read a bit of the landscape. We also have about half hour left. We got to come up.
I think I can get 20.
That still gives us 2.5 hours.
2800
divided by 20.
On deck at 4, right?
Yeah.
So off bottom at
60. 2.3. Yeah, I mean, you could get another. Let's see. 10 minutes out of it.
The good news is we haven't filled your vehicles with rocks, so dropping the ballast will do something.
Well, it's not too late. There's rocks here. So half an hour. Winchy up, right?
Yeah, half hour.
Roger.
Doing any Niskins on the ascent?
I was actually just about to check our sample list and see what we've got left in terms of Niskins.
We've got 2 left.
2 left? Okay,
Yeah.
that's right, because we fired, so might fire 1 right off the seafloor and then 1 at 700 meters.
700. Is there got an incline there, or what do you call it, where the stratification?
I just know it's one of the standard depths that some of the groups that are doing POM usually use. And I don't know why they chose that particular one, but it's what I'm used to seeing on their list, so I just try to match it up to there. I don't know that it's specifically a magic number.
They're like, we
Or
were just
they'll do,
doing it
like-
because you were doing it.
Yeah.
Oh, that's a big bathy something. Pathy something.
Oh yeah.
That's almost there.
He's looking a little sad.
Yeah.
Yeah, there've been a couple of them that have looked a little rough. I know the one that was sampled was missing most of its pinnules right in the middle, so we use that as a convenient target to clip.
All right, that's the end of transect.
All right.
And
Find some rocks, Jake.
free to explore around.
We want rocks.
We'll
Yeah,
still be-
we want rocks.
Ooh, a little spicy on that. Argus heat.
Yeah, got a little low there. The freaking big swell got me.
Yeah,
I was trying to time it.
I've been watching the wire cam and it looks like we're getting close, but it doesn't appear that we're, plus now that I stopped, there's a lot less of that tension.
I think you'd have to move it 10 knots for it to hit the ship at this depth.
Well, not in the heaves. It's kind of really, it was pretty close out there, but I think,
Oh, that's right. That's because our tail sticks out the wrong way. That's
Yeah.
going to so not be an issue after we get another 12 meters on the back of the boat.
yeah, apparently they're going to change the angle, which would be great.
Yeah, it's a great angle if you're hauling nets up or something, right?
Yeah.
So how much swing do we have left before we need to move the ship again, or do we think we'll just be moving through the water till on
Well,
the last thing before the pull?
we're, yeah, we're just going to keep swinging.
200 meters?
Yeah, about 200 meters, 150 to 200 meters.
Okay.
Auto deploy.
Yeah, and then we'll see. I mean, I could move the ship now, but I'm not really quite sure what direction. We could go northish, but by the time we get up there, perhaps it will probably be time to pull anyway. So see what kind of terrain we have, and then it's
I think
while it swings.
by the time it swings in, it's going to be time to
Yeah.
pull the plug.
Right.
Okay.
Or well, you'll have maybe 10 minutes to play around there,
Yeah.
10 or 15.
So this slope here did yield some outcrop here.
Yeah. I'm hoping we run through another one. Guy Cochran, our geologist ashore, is hoping to see some more rocks before we have to take off. So
Does he
looking
want
for
any of those
options for
rocks?
him. I can ask.
I guess I should see Taylor Ann. What kind of stuff do we have?
In terms of samples,
Yeah.
we have a lot of free room still, but we do have some floaty things in both sides of the boxes, so forward and starboard.
Yeah, I'd hate to lose those at this point. For
Yeah.
Yeah.
But I guess if
Picking
you
up a
were to
sample
go
of
for the
opportunity.
starboard box, you could just pull it out like halfway, because we do have one big box that's free, but A has something floaty in it.
The good news is he says he doesn't want any rocks.
Okay.
We just want a visual survey of them.
Yeah.
yeah, thanks for that. It's be tragic to lose a sample we've been tacking around for 20 hours.
I'm
Yeah.
excited to see the one in the slurp jar 3. What was it? It's like the octocoral with 3. It's like
Oh, the Bathyalcyon? With
Bathyalcyon.
Just the 2 giant polyps.
Yeah.
I'm also excited to watch the unload from the camera view, unless I fall asleep trying to watch them process samples.
You're getting
Hey,
a little
this is
far
Lisa. I'm
away.
back.
Hi, Lisa.
Come,
That was
come to your
hi.
left up the hill.
Yeah. I was watching while you went through that last rocky outcrop. That's cool. So for those of you who are watching, I was just on a call with the other watch leads, and our next dive after the Santa Lucia escarpment is we will be going much further near shore, and we're going to be checking out a petrale sole, which is a type of flatfish, and we have done dives in that area in the past using an AUV, an autonomous underwater vehicle, and they observed petrale spawning there,
Backing down.
petrale sole spawning.
Yeah, you have to go. Sorry, you have to go closer to the ship. So whatever you got to do that. Down maybe.
Down.
That's what we did yesterday and it didn't work.
There's some of our mud dusting off the vehicle.
Probably remnants of my crash landing last night.
Every time you just park on the bottom, we have that fiberglass grating to protect all the gear under there, but it's a really good cookie cutter.
What if you replace that with something solid, like a solid panel? Would that make flying difficult? I guess going up and down may.
Yeah, so what the cool kids have done now is they've taken like a 1/4-inch sheet of aluminum and cut basically 100 millimeter or 10 centimeter or 4-inch squares in it.
Mm-hmm.
And then they've milled slots in between those squares and then had in all the corners a bolt hole. So you have this beautiful piece of aluminum.
Like
that
a
you can
pattern of.
Yeah, you can reach your hand through it,
Yeah.
and you can mount stuff on it and slide it around. One of the kids at Schilling came up with that, and then Sebastian copied it. It was the first one they did. They polished it, right? They milled this beautiful piece of aluminum out and then spent hours and hours polishing it. It was like a mirror, and then that was the show vehicle that had the Corvette paint job on it, right? It never went anywhere except I don't even think it went in the pool. But one of the Schmidt guys was up there and saw it and liked it, so he made the same thing on Sebastian. It's beautiful to be able to mount science gear on, but it lets all the water flow come through. So the reason for the grate is it's easy, it's very structural, and you get the water flow through it. If it was solid, it would be like our starboard mailbox, which is
Liability on ascents.
yeah. As Bob says, moving a piece of plywood through the water.
Yeah.
The wrong way.
I think we once asked, like 2 years ago, as we were doing some dive planning, just how many Niskin bottles could be mounted on the Hercules. And it was pointed out that many could in theory, but the more you put on there, the more they just act as a sail anytime there's a current.
Yeah.
Yeah, we call where the Niskins are currently mounted, we call that the anti-maintenance fence.
Yeah, you can't reach anything.
It's this row of Niskins on some aluminum angle all welded together. It sticks out the port side.
See it on Argus. Sticking out. So it ended up being quite steep here.
Yeah. Now we just need to add a few more rocks.
Yeah. Well, but not steep enough to not bury the rock that's there, or the sort of shelves that I think are there.
There's a
Oop, there's a, yeah.
Oh, that's a really nice coral.
Maybe bump him to the north a little, Rennie. It looks like.
Yeah. Rich,
get a
now.
few more minutes
with the few more minutes that we have, we don't need to collect anything, do we, Meredith?
No, I was gonna have them
Okay.
fire a Niskin right
50
before we
meters
leave
north.
the seafloor, and
Okay,
then one on the way
great,
up. We can just survey around.
good. A little relaxed then. So what kind of coral is that we, can we zoom in on it?
Right at the end of my leash.
Here, let me,
Oh, okay,
I'll flip around.
that's okay.
You can
Might
pull
have
it,
a,
pull it around. Pull on it. You can drag Argus like 5 meters easy.
Yeah.
Happen to be on a good heading there.
drag it right into the wall.
Yeah, we've dragged Argus quite a bit at depth on ONC.
It's fun, Jake. You get to use full power.
It's kind of crazy when you stop the forward thrust.
It makes it really hard to fly the vehicle because
Yeah.
Right.
I can
But it's
see that
quite
right now. Yeah.
you can do it. You can do it.
Yep, I think that's another Primnoid.
That's
Okay.
where the
Just another.
Yeah, just another. One of
non-digital
these really rare,
keep applying force
really.
or thrust.
Keep applying force.
Keep your vector, Victor.
One of these rare species that we never see unless we're this deep.
Ah.
So
I can come.
Well,
Let me
we-
come down a little too. Give me out.
we've gotten jaded
it
in
looks-
24 hours.
Yes. So it looks like it has those brittle stars that are pink that are associated with it
Yep.
sometimes or
Yep.
often. Yes.
Yeah, it's got a few brittle stars, using it to kind of get out off the wall into the water column, or just hanging out around the base now, but I bet they climb out there.
So this is an example
Quite
of
steep here.
how corals provide a biogenic habitat for other species like these brittle stars that will use it to, as Meredith just said, they'll climb out on the coral stalks and they will extend their arms out into the current. So the
Actually,
coral gives
I
them
was-
a better perch.
Butt cam is the new Argus HD.
Yeah.
You can see.
Yeah.
Just
Okay, try again. Should have enough leash now to be a little more comfortable.
And what species of Primnoid is it, Meredith?
can
I think this is Narella.
Oh, right.
And I'm not sure which of the Narella species it is.
go for a zoom there, Steve.
Sure, it looks sort of lacy, but it must have
Yep.
some
There's a line caught
rigidity
up
to
in
it
it.
because
See that at the bottom?
oh yeah, good eye.
Oh yeah. Getting in.
Where'd that come from?
Oh, yeah.
Yeah, all the way out
Oh,
I
here.
thought that was biological.
Somebody tossed it over the side of a vessel.
Yeah,
That's where it landed.
it's like a long time ago.
Oh, you can see one brittle star is sort of hiding in the branches there.
Yep. Up on the fronds, just over the right-hand side.
Yeah. And there's a crinoid tucked in there by one of the brittle stars, or maybe that's the brittle star itself, but
There is a crinoid up top.
yeah.
Yeah.
I think.
Hey, Steve, put Argus butt cam there.
I can't put it there, but I can put it on 3.
Oh, that's hardwired, is it?
Well, it's not hardwired, but that's
Yeah,
It's
part of the
recording.
deliverables that we keep that there.
Oh, because that's what's recorded on shore?
Yeah.
What's that pink thing in the bottom?
The bottom right.
Yeah.
yeah.
Oh, that's the sea
Put it
pen.
on 3.
Oh, is it?
Yeah, that's a little sea pen.
So Steve has put Argus's aft camera up on the satellite feed 3. You can
Yeah.
see it
Yeah.
whipping Herc. That's what Jake's dealing with now.
Yeah.
Or on par for, you can barely see Herc in the background back there. Tail to tail, we call it.
You'll notice on the
Definitely a challenge.
next dive when it's only a few hundred meters of water how much we can just put Argus wherever we want, whenever we want it. We're not dealing with all these time
Right.
limitations of being in the deep and swinging around.
Yes.
Yeah, you can zoom.
The
Sampling on
next
fly.
dive is the next dive will only be 500 meters. So
Jeez, it's barely enough to get Herc wet.
yeah.
The kiddie pool.
Is it worth
Cool.
it?
We'll stay in the shallow end.
So which sea pen is that?
I think this is Penatula. It would be
Oh.
Penatula phosphorea.
I think that's the first one I've seen on this watch, or did we see them before?
On this watch, I think that's the first one. I thought I saw one just as the last watch was ending, but we haven't really seen them. I think we're just getting into the depth zones where they start.
Mm.
They don't tend to go super, super deep. These are really cool little species. They're one of the first described species, and they're bioluminescent.
Really?
And even that's where they get the name phosphorea. A lot of the sea pens are bioluminescent.
If we turn off our lights, can we see them?
I think we tried that before and we ended up need
Lost
a low
in
light.
the dark.
Yeah, need a low light camera.
Yeah, some of them you need a low light camera for. And because I've tried it before with Amphiptyllum, because that one's also bioluminescent, and we weren't able to see it, but they apparently do flash blue.
Probably see what else we got.
And this guy, you'd also have to usually you have to touch them and disturb them, so we'd have to get set on the wall and then disturb them, and they'll also usually only flash once or twice that I'm aware of in the sea pens.
Blue flash.
Yeah. So this zooming in with the camera that we're doing, I think is just a great example of as you're just flying by, it seems like there isn't much going on, but when you look up closely, there's actually quite a bit of biodiversity and quite a few animals living on this what seems to be desert seafloor.
Yep, all kinds of stuff.
You might get enough lace to come up the slope again. Maybe. Or you can, if you back away a bit, you might pick up some rocks to look at. Shouldn't have to back away. Should just let go of that stick and it'll rubber band you back.
Herc's driving itself.
Yeah, some of the bamboo corals bioluminesce as well. There's a great video from an ROV out of Hawaii where they used the manipulator arm and grabbed the bamboo coral and then turned the lights down, and you can see these like pulsating blue waves of light go up the skeleton.
Cool.
I can't tell in Argus's tail camera, but there might be something down below.
Yeah, another bit of ledge there. You can see shadow and Herc.
Yeah, it does.
Herc's athlites.
Go down a bit.
Yeah. Down and back.
Lower should get us closer to the ship too.
Yeah.
My ulterior motive.
One of our scientists ashore is pointing out that there will be, even if you scoop up one of these mud samples, there will be a huge number of what they call infaunal taxa that are all the little microbes and microinvertebrates that are found in the mud itself.
And is
That's
the environmental... Oh, yeah, go ahead. Maybe that's what you were going to lead into.
I was going to say, it's one of the reasons they like to take the sediment cores.
Okay. So would the environmental DNA work, would that be something that could help elucidate or help us figure out what is living in what these infaunal organisms are, because we can't really see them?
I don't know if you'd pick up how much of the infauna you'd pick up from environmental DNA. Certainly, if you took like the water sample off the surface of the mud, it would probably help. But up in the water column, it will just really depend on how much water exchange there is through that fine-grained sediment. So you'd get
Right.
some, but I think you'd have to get the water sample, or what some groups do is actually filter out the mud and then you can
And then
use
take
that
the,
water.
yeah,
Yeah.
right. Because they're shedding, just like all animals do, but it's probably concentrated around the sediments that
Yeah.
they're in. It's
Rocks
not,
back in your
yeah.
archive?
Rocks below you.
Yeah.
Yeah.
Crispin says, more nematodes than you can shake an ROV at. Yeah, when we take the environmental DNA samples, we're not usually pulling strands of just the DNA itself. It's usually, we suspect, cellular material that all the animals just shed into the water. And it actually is, when we're around certain types of animals, we have to sometimes make adjustments to our chemistry. So like the bamboo corals, especially the big Isabella fans, they're like mucus bombs. They just shed tons and tons and tons of mucus into the water. And so when we take a water sample around them, they can just dominate everything in the area, even if there's only 2 or 3 of them around, just because they're putting out so much more cellular material than the other animals.
Zoom there, Steve. What is that little pink guy?
Is that a mushroom coral?
I think that's a little mushroom coral.
Tiny one.
Yeah,
And
you
as
guys
we
have
were
such a good eye.
We also have giant monitors.
Yeah.
Yeah. Thank you for helping clarify that, because I'm sometimes thinking like, I need to go to the optometrist again.
Giant monitors, and we're a couple seconds before you, I think. So.
And we
Okay.
have different, even bigger monitors than when you've sailed, if you haven't sailed this year.
Don't give away our secrets or anything.
Yeah.
Yeah.
Your superpowers are diminishing.
Yeah.
I think there's some.
For this
sheer
watch, I got to move
below
out
you
to my
to your
living
right.
room where I have a big
Yeah.
monitor.
Come, what, Steve?
We can even take all 4 of these monitors, the big ones that you see in Sat-3, and we can make it one image.
Oh my.
Yeah, we've got to see that in operations.
So in a way, you do have a microscope right there. So there is that the Isabella right there?
So that is a
The
bamboo coral, but that one is Lepidopsis and not Isabella.
Okay. Okay.
The long white one?
Yeah.
Down there?
Although I will
Great
say
Argus
that
shot.
is
Yeah, it is.
somewhat of a hand-waving identification, because Isabella does start life as a single stalk before it starts branching. But we've been seeing a lot of what are definitely Lepidopsis, because they start to curl at the top, which Isabella doesn't. And so I'm pretty confident that most of these are Lepidopsis.
There's the big heave there.
Yeah.
Ooh.
Yeah. The wire's a lot happier now that we're not
Yeah, it is.
pulling back.
Is that a little squat lobster?
Yeah. Like it.
Fly in till you touch, Jake. Just ever so gently.
That seems like bait to get me to crash into the wall.
Yeah.
Now, what are those things down
8
below
minutes to
under
crash.
that? That little shelf there with white thing.
Down?
Yeah. You see that?
Yeah.
What are they? Case in point of
Oh, yeah.
on stuff and getting closer and closer, you see more.
Zoom there, Steve.
Oh, yeah.
Good eye.
Oh, this is more of those benthic ctenophores.
Oh,
So that sponge that we saw that had stuff all over it,
Mhm.
like with the weird streamers coming off, that was apparently a benthic ctenophore that had settled, and this looks like more of them just pointed down instead
Huh.
of streaming out through the water column.
I would not have said that those were ctenophores. It's amazing. That's so different from what you see in the water.
Yeah.
They like tether themselves with a little, or they just sticking out.
Unless I'm wrong, those might just be hydroids now that I'm getting a really close look at them.
Yeah,
Yeah.
different form factor of hydroids than I normally see.
Yeah. I mean, they're definitely jelly-like and benthic. Let me look at my picture that Gary sent me. Oh, they could be this, but they could be benthic ctenophores looking at the pictures that I'm looking at right now.
All right, this last 5 minutes, we'll have to, you want to trigger a Niskin at the bottom?
Yeah, let's go ahead and trigger one last Niskin here, and then we'll trigger one on the way up.
Sure.
You wrecked all the way back, are you?
Nope. Coming back.
Kept the craft in pace with the camera.
Yeah, which color am I going for here? Look left a little, please.
So we have the last 2, 5, and 6 are open.
Green.
Yeah.
No, white.
I think green is 5 and white is 6.
Yeah, so I want green.
Yeah.
Okay. You can rack back out.
What was that sample number?
07272.
072. 5.
Hopefully there'll be some Chrysogorgia DNA in there along with the couple of bamboo corals that are around. We were headed down this ledgy system. I did see one little Chrysogorgia. They are around.
I'm gonna leave that thing hot for the trip up.
So the waypoint that we're at, or that we've passed, because we're not really doing the waypoints anymore, which one is that, just for my records?
Sure, let me zoom out. We were between 5H and 5I.
Great. Thank you.
Closer to 5I. I don't remember those being, were those added?
I
Oh no, we
think
had those as spares, right?
it was there. It actually goes to 5J,
That's right.
but we were hoping to make it at least to 5H, which we did.
Yeah, so we got some bonus.
Yeah. Some bonus cliffs here.
Yeah.
Priming the winch
Yeah.
auto.
Yeah, let's get the winch auto. You gotta prime it.
One glory shot of Herc there.
Yeah, that's really cool.
Nice last little rock outcrop before the blue water.
Yeah.
Yep.
It looks like you got a, oh, no, you're all right. Let's see, which way? Yeah, you're all right.
Oh, can we focus in right at the bottom of the ledge here too, if there's time, or if we're just climbing up that?
We got
One last one, and
One
that's
last one.
it.
Where at?
Pulling the plug.
You're almost there, just a little bit more. Oh no, it's okay. I thought there was something hiding there.
oh,
Never
there.
mind.
Yeah, it's a rock or something tucked in there.
Okay, winching
Yeah.
up.
Winching up,
Okay.
All right.
flying up.
Thank you.
Goodbye, Santa Lucia Bank Escarpment.
Yes.
Herc's gonna get a Yankee soon.
Good up.
Hope.
I try and hold out till the last second.
Yeah.
Coming
Hopefully
up
it
at
won't be
25
so long.
meters a minute. I come up at 15 first till we clear the. Stay on the cliff as long as you can if you want.
Great.
Let's see, I need to come around. If I come to my counterclock, I don't think
Ship's
it'll
heading
turn
is
out, right?
310, and I suspect it'll be somewhere around there for recovery.
I have 0.7 reps.
See, so that's going the wrong way. I need to go the other way. I'll wait till we get off that. Till we get blue water to do that. Testing.
So it's about 1:30 now, and we think the ascent will take 2 hours or so.
2.5 hours is what we're calculating if we can make 20 meters a minute.
Yeah.
Okay. And we'll be reviewing and seeing a completely different community of marine animals that
Just
live
keep
in
flying
the
forward there. I'm going to come around.
mid-water.
Yeah. What'll be interesting is since the descent, well, I guess the descent was about 4 p.m. So if we had a bigger time lag, we might have been able to see a slight difference in the vertical migration that happens every day. But I think that we'll encounter the same animals going up as coming down at about the same time.
Right.
So you want to keep some forward on the whole way now?
Yep.
Pull the tether tight. Try and hit the cliff.
Bacterial
There you
mat in the background right as we're leaving.
Yeah.
Yeah.
Pull on it, Jake. Pull.
And a black coral also. Bye, black coral.
Yep.
Okay, going to 25.
The way that we're moving up, you can really feel that it's being wenched up.
Yeah.
Yeah, he's getting winched, all right. Sorry, 20. That's what we said, right?
20, yeah.
You'll have to go 100% joy gain now and Kill your Z bias.
Let's see the winch in action on
Press the little
feed
button
3.
there. And cue about 20% forward.
20% forward.
Just 20%.
Oh, yeah.
Yeah, lock that in.
Stay off bottom, yeah.
Did a little too much.
So what are you getting there? We're getting, yeah, getting 22 at the moment. Sweet.
Ooh,
Slow down
that
till
was
you
a
catch
yank.
up there.
Yeah,
Yeah, sorry. No, you're good. It's
the heave.
all the
Yeah.
winch now. I got too far above you.
I sorted out my extra big pseudo fish guide for the ascent, just in case we see something cool. I can try to quickly flash over to it, but I don't know if I'll be able to flip there that fast.
Yes. I'm concerned about that as well, but you never know.
Yes.
I'm gonna make that all blue water for him. Screen, you can pan it. Tilt up till
So
you're
all
just
that,
in the
yeah.
up. Yeah, somewhere in there.
Thanks for that.
All those white flecks that we see raining down are appropriately called marine snow. And some animals feed on marine snow. A lot of the ones that we saw on the benthic sea floor.
Pull your utility page up, please.
Yeah, down there, pretty much anything that's not a predator or not living in a
I'm
chemosynthetic
gonna drop
community
away.
is dependent on this.
All right.
You're full up, right?
And a huge
Yeah,
source
so
of carbon.
you want to give
Yep.
less
Less vertical?
forward. No,
Less
full
forward, less forward.
Yeah, see how you drop down to 2,000 PSI?
Yep.
Sorry, we're going to steal your camera here to drop a weight.
Oh,
Show me
that's just
the
fine.
show me the knobby there.
So yeah, the marine snow is just a lot of animals that have died in the in what we call the photic zone, where you have phytoplankton
Okay.
that
really
use
coming
the energy of the sun.
Can put
They're
the
the
camera
primary
back up for 'em.
producers. And then they lead to the feeding upon them by zooplankton. So a lot of this are zooplankton that have died.
Mm-hmm. Some of these will actually be clumps of some of that phytoplankton too that have all kind of glued themselves together and gotten so heavy they're no longer buoyant and will start to drift down themselves.
Yeah, and then a lot of the sea cucumbers that we saw at the seafloor, they feed on this.
Yep. And hopefully while we're in this blue water transit, we'll get an opportunity to see some of the midwater filter feeders that feed on this as well, particularly the larvaceans and their big mucus houses. It's a good area for them.
And we saw also the mucus houses themselves become marine snow because they will discard them, and then we saw some of them also while we were on the bottom, on the cliffs of
Yeah, I think
the
we
escarpment.
actually saw one or two like wrapped around corals down there.
Yeah.
I'm going to pitch the other one.
So the pilots are currently dropping some of the ballast weights from the ROV. This helps the ROV stay just slightly positively buoyant when it's on the bottom, but now that we're ascending, they'd like to make a quicker ascent. So by dropping these, we'll be able to ascend faster.
Okay.
Didn't Bob say something about Mongo being?
Yeah, we'll deal with that when we get close to the surface.
All right.
Show them they want to see the blue water. I'm gonna turn this guy off. I think it, yeah, look at that.
Is the porch light on?
Probably.
A little bit.
This much marine snow in the water is actually a sign of just how productive this coast is. We've got the cold California current moving down from the north, and the warm water current moving up from the south, and all of these currents are bringing up this cold, nutrient-rich deep water that allows the phytoplankton to bloom on the surface and start this really enormous, rich food chain. Got in areas of the Central Pacific, they're occasionally referred to as marine deserts. You can have this perfectly crystal clear water that goes all the way to the bottom. You can see for hundreds of meters, there's not nearly as much marine snow because the water just doesn't support as much plankton and therefore doesn't support quite such the food chain that you get out here on the West Coast.
Yes. It attracts migrating animals, birds, and marine mammals, whales come to this area because of the incredible productivity that is pretty consistent seasonally from year to year.
Yeah, you can see in the sanctuaries on the West Coast a lot of like the big whale species that come through. I think I've seen blue whales in Olympic Coast National Marine Sanctuary and in Channel Islands National Marine Sanctuary, which was really cool.
Yeah, with the research that Point Blue has done, they modeled that the hot spots of productivity and forage fish and krill, they coincide with the locations of national marine sanctuaries where you have these birds that fly from all over the world and whales come in to feed in the productive waters.
That's excellent.
Yeah. And
one of the few
No, go ahead.
I was gonna say, it's one of the few disadvantages of broadcasting from your living room is, you know, when you're not on watch and you're on the ship, you can go outside and whale watch and bird watch.
Do the folks when they're not? Ooh, somebody's dog. Hello. That's
Intruder alert.
that's the
Intruder alert.
yeah. So I think Meredith, that's her dog, not mine. So she said that somebody might come by. So I'll narrate a little bit right now. Well, or I'm going to ask a question of the Nautilus crew. So when you're not on watch, do some of you have then the opportunity to go out and whale watch or bird watch? And somebody saw a white shark, so shark watching.
Yeah, we've had some a lot of dolphins on this cruise and a few whales we've spotted. I saw a shark as well. So yeah, we do occasionally get time to do that. It's nice.
Yeah, you've had nice weather the last couple of days from what we could see from the different cameras around the ship.
I'm back, but I'm hovering over the mute button.